Thank you, Ray, for the introduction. I would like to thank also Adam and Harry for the kind invitation. It's always a pleasure to be here. I will give you an overview of what is going on in terms of gene editing, but also on epigenome editing. So I enlarge a little bit the topic today. So here are my links of interest, and I would like to acknowledge right away the different companies who shared data, so Chroma, Excision, Precision, and Tune. Here, I mean, one of the key points here is to see how we could cure HBV infection with antivirals. I will not touch on immunotherapy. You've heard about it earlier on. And we've been, as you all know, been mainly developing in the last decades or so strategies to suppress viral replication with direct-acting antivirals. But the issue is that the viral reservoir is very difficult to eliminate or silence. And we are talking about either the episomal form of the virus, so cccDNA, or the integrated forms. And in that respect, so we can envision strategies either to silence cccDNA or integrated viral DNA, or trying to eliminate the viral genome reservoir. So in that respect, I mean, we have seen different small and large companies who have developed screening programs for drug discovery of small molecules that would target cccDNA, but finally failed in that endeavor. And what came out more recently with all these progress and discoveries made with the CRISPR-Cas9 technologies is that gene editing could be a very interesting approach to target the HBV genome, and more recently, the epigenome editing could be even an additional opportunity. So, as you know, we've been evolving from the CRISPR-Cas9 technologies to the more refined base editing, and then there's also the Arcus technology that we'll touch upon, and then there's this epigenetic editing that I will cover in a few minutes. Regarding gene editing, so we started first with the CRISPR-Cas9 nuclease technologies, which induces, as you can see on the left-hand side of the slide, with the Cas9 nuclease double-stranded break that will generate through non-homologous end joining DNA repair some different either insertion, deletions, mutations in the targeted genome, and with the advantage that this approach, if you use the right guide RNAs, can target both cccDNA and integrated sequences. And we may see either a disruption of the HBV ORS or even cccDNA destruction. But we know the caveats with all these technologies, and I will come back to that in a second. And there were newer versions of gene editors that were developed, and mainly with base editing, where you can use a similar technology where you, with using guide RNAs to direct your gene editing technology, using usually a dead Cas9 that is combined to a deaminase, so that you have a specific deamination of either cytosine or adenine. And in that respect, you create a single point mutation without doing double strand break in the targeted DNA sequences. And you can target both cccDNA and integrated sequences, and you can induce mutation in those targeted sequences. And here you see what was the main issue with the CRISPR-Cas9 nuclease in the editing outcomes, although there are ways to reduce this risk, and this risk is a chromosomal rearrangements, and this may lead to chromosomal recombination when you target not only cccDNA, but also the integrated sequences that share the same viral sequences. So this is something that we need to keep in mind when developing this type of strategies. But on the other hand, we are also learning a lot from the gene editing field. And as you can see, quite a few number of clinical studies, not for HBV, but for gene-inherited disease or other type of disease where gene editing is currently in clinical trials. So now they're at the time of the last summer, I mean, there were more than 140 patients that were that have been entered in gene editing therapies. So for different indications that you can see, but so we will learn from these studies in terms of delivery efficiency, off-target effects, and so on. So this will be extremely useful to inform us for HBV and potentially HDV treatment. Here I just want to show you a couple of slides regarding our experience on gene editing. So we moved from the classic CRISPR-Cas9 approaches to the base editing, and we showed that with the C-base editors, where with that technology, you can introduce stop codons in cccDNA as well as integrated HBV DNA, that we could induce a sustained inhibition of HBV replication and HBs antigen production in primary human hepatocytes as well as in mouse models of HBV infection. However, there are some caveats with the C-base editors, and there were newer versions of deaminase and base editors that we were working on with in collaboration with Beam Therapeutics, and with the adenine base editors, and we can induce missense mutation, and we targeted the HBS open reading frame with 2 different guide RNAs that were highly conserved among the different genotypes, and we studied them in the different HBV study models. And we showed that in the cell culture system, where you have the active and full replication cycle in the HBG2 NTCP cells and in primary human hepatocyte, that you can achieve a profound decrease in HBs antigen expression and secretion, and also same type of effect in cell culture systems where you have integrated HBV, either artificially integrated in the HBG2-15 cells or naturally integrated in the PLC-PIRA5 cells, so you can achieve a significant reduction in HBs antigen production, which is something really interesting. And we confirm the data in vivo in the HBV mini circle mouse model, as well as in liver humanized mice, where we observed quite nice and significant HBS reduction in the serum of these mice. And then we were interested to see, since this was a quite consistent reduction of HBS antigen expression, whether this could be helpful also in the context of HDV infection. And we showed with the same approach, so using the PLC-PR5 cells, that the decrease in HBS antigen expression was associated also with the decrease in HDV RNA release when the same cells were transfected with HDV RNA. And then we showed that the cells supernatant from these cells were less infectious in primary human hepatocytes. So this approach could be very interesting, not only for HBV, but also for Delta infection, as we could suggest that this would decrease the release and the spread of HDV. So this is something that needs to be kept in mind for the future. Now, in terms of other gene editing approaches, a few slides from precision bioscience using the Arcus gene editing technology, where here the Arcus is delivered through LNP, and it recognizes the highly conserved sequence in chronically infected individuals. And here, what is important is that you don't need a guide RNA to target the HBV genome sequence, and you can target both the CCC DNA and integrated viral DNA. And they did quite a lot of preclinical studies to assess antiviral efficacy and the safety. So they mainly used a AAV a non-human primate model to show that it could reach 99% viral DNA editing. In primary human hepatocyte, there was a strong effect on CCC DNA. And in the AAV mouse model, there was a 95% reduction of HBS antigen that was durable. And they also showed a reduction of HBS antigen in transgenic mice that harbor a fully replicative HBV genome. And there was a dose-dependent editing on HBS antigen reduction. Regarding the safety, a lot of studies have been done preclinically and showed so far no increased risk of translocation or integration. It was well tolerated in non-human primates, and the mRNA and the LNP were cleared rapidly after each dose, and there was a transient transaminase elevation in these animals without any other sign. And now the precision is ready to go to the clinic, so we are looking forward to that. So it's quite exciting. So the other editing strategy from companies comes from Excision Biotherapeutics. They were showing their data at the HBV meeting a month ago in Chicago. Here they use paired guide RNAs to target two sites in the HBV genome, and they can target both integrated and CCC DNA to lead to deletion, degradation, and viral inactivation of the genome. And here's a summary slide of the antiviral effect that they observed in the AAV HBV mouse model with an important reduction, 80% reduction of the intrahepatic HBV DNA with the highest dose, which subsequently led to a decrease in HBs antigen levels in serum by 97%, so 2 log decline with the higher dose, and also a 2 log decline in HBV DNA in this AAV HBV model. So quite interesting, very nice results, and we're looking forward to more results with that technology. As I told you, there's also another approach, which is not to induce mutation in the HBV genome or its degradation, but the other approach is to silence the HBV genome using epigenome repressors. And here in that strategy, the idea is to induce an epigenomic editing to inhibit HBV, and the main strategy here is to use a dead Cas9 that is combined with a methylase that will then methylate the DNA, and using the right guide RNAs, and you can target the right sequences and methylate the CpG island on both cccDNA and integrated sequences, and induce an epigenetic repression and inhibit HBV transcription and, in the end, replication. But one of the caveats is the potential for off-target effects of the epigenomic editing with unwanted methylation of host genes. So this will have to be extremely well evaluated in preclinical models before going to clinical studies. And here we show you two examples from Chroma. And Chroma is using this type of approach, using here, you see here their construct with which use a methylation effector domain as well as a transcription effector domain combined to target the cccDNA and integrated sequences. So they have a very efficient silencing of both cccDNA and the integrated forms. It's durable, and it is so far specific and preserves the genomic integrity. So here is a summary slide that they presented at the HBV meeting in Chicago, where they showed a 3 log decline in of HBs antigen in the AAV HBV mouse model with a single dose. And they, with the immunostaining that they performed in the liver tissue of these animals, they showed quite significant clearance of the number of infected cells harboring core antigen. And it was highly specific so far with no detectable off-target changes. And they showed also in non-human primate that it might be translatable to the human situation using the PCSK9 guide RNA substitution. The other company that is doing also very interesting work in that area is Attune with an epigenetic silencing approach, trying to, again, with the same type of strategy, obviously with their own specifics, to target both cccDNA and integrated sequences. They showed very nicely with a single liposuction a very high CpG island methylation in the targeted region, as you can see here, that would enable a long-term epigenetic repression of all viral transcript from a single dose. And you see here results in using the HBV RNA analysis, and you see a strong decline in HBV RNA transcription compared to the control experiment using non-targeting guide RNAs. Here you see results in primary human hepatocytes with a very strong decline in HBV RNA, and in humanized mice, a decline in serum HBs antigen by more than 60%, which was the theoretical maximum repression that they could observe in this model. So altogether, I mean, we have really very promising approaches at the preclinical stage, and we are really eager to see what will happen in the clinic, either with the gene editing or the epigenetic editing approaches. So to conclude, I think we have these very exciting approaches to target both cccDNA and integrated sequences. They have the potential for a permanent inactivation or silencing of the viral genome. Obviously, there are many questions that remain to be addressed. So as you've seen, there's an heterogeneity of the study models that has been used by the different groups: transgenic mice, AAV, HBV mouse model, liver humanized mice. So the question is whether we will be able, with those technologies, to target the real cccDNA in patients, the real integrated sequences, the real infected cells, but that's the purpose of clinical trials. There are also issues regarding delivery efficacy, infected hepatocyte coverage, viral genome accessibility, depending on the epigenetic regulation of the genome, and we'll have to see in the first-in-human trial that will start very soon. And we'll really, on the safety side, we'll have to be extremely cautious regarding the off-target effects on the human genome and the long-term safety prediction. But there's, as I said, I mean, it's a very rapidly evolving field with constant improvement in technologies, new versions of nucleases, guide RNAs, delivery systems, and so on. So we'll need to keep the innovation going on and adapt to that. Again, just to remind you that this is not only a Star Wars science. It's now in the clinic. There are clinical trials for different disease indication, and we will learn from that. And now the real question will be clinical studies. We have challenges regarding patient population, study design, endpoints and safety criteria definition, long-term efficacy and safety assessment. But this is really where we need to go to drive the science to help us to go in the direction of functional cure for HBV. And I would like to acknowledge my collaborators in Lyon working on gene editing, as well as collaborators with whom we had scientific discussions regarding these different approaches, and I would like to thank you for your attention.
Okay.